This is one of the pictures of him out in the field with his giant pile of Piezaster on top of a mussel bed. And a lot of his research was looking at the interactions between sea stars and their environment and overall just diversity. But it really led down this path of the role that Piezaster plays in shaping the intertidal community on the outer coast. And I just want to spend a little bit of time going over two major concepts that Bob Payne coined specifically from Piezaster to sort of help frame this organism as I start getting into some of my own work. So the first is keystone predation. And this says that a predator increases diversity by removing a competitive dominant. So if you take a nice little rocky intertidal shore here, you add a whole bunch of mussels, a nice big mussel bed, and mussels, when they establish, they've got all these little threads. They use that to hold on to the rocks. They use it to hold on to each other, and they can basically sit and grow on top of whatever's already down on the rocks and come out with this nice big mat of mussels and outgrow anything else that's out there. So this would be considered your competitive dominant organism. If you introduce Piezaster into the situation, Piezaster loves mussels, so they'll just eat away and gnaw at them all the way up to the point where Piezaster can't really handle the heat and the desiccation on the intertidal. So you end up with this really nice band of mussels. And this area below it that's devoid of mussels, mostly because Piezaster is going in and eating it. And when Piezaster eats all those mussels, it opens up this nice rocky space for things to grow that couldn't be there before because of the mussels. There's been some back and forth about this more recently in terms of whether or not Piezaster is actually increasing diversity, mostly because there's lots of little things that grow within the mussels. But nonetheless, they're at least changing what's out there on the rocks and changing the community in general. Another way to just visually see this is that the top predator is eating the dominant competitor. The dominant competitor is out competing the weaker competitor. The next important thing is keystone species. So similar in phrasing, slightly different in what it means. And this one says, this one applies to any species with an effect disproportionate to its biomass. And here's a really nice graph that I love to help describe this. And we'll sort of start over here with the dominance. If you picture something like a single tree in a field, that single tree might bring in a couple new birds and a couple new bugs, but overall might not have a huge impact on the overall area. Whereas if you had hundreds, maybe thousands of trees, now you're talking about a forest. It might be affecting the way the sunlight comes in. It could be changing the way the wind is moving through it. Ultimately, you have changed this habitat and you've got a lot more niches and little places where things can hide. So the dominants are things that you have in very large numbers, and when they're there in large numbers, they have a huge effect on the habitat. So trees, kelp forests, even mussel beds could fall into this category. On the other side of that, you have your keystone species. And these are organisms that have these really big impacts, but it doesn't take hundreds or thousands of individuals to make this impact. It takes a much less, far fewer of them to have this impact. So with the sea stars here, you have lots and lots and lots of mussels to make a mussel bed and to change the environment of what's happening on the rocks, but it only takes a handful of Piezaster to have this kind of effect on the mussel beds and to open up this space. So Piezaster has been shown to be both a keystone predator and a keystone species. And this is really important because, you know, this is one of those things that really kicked off research in sea stars. There's been many, many people that have come up after Bob Payne to get more into the nitty gritty of how Piezaster is interacting with things in their environment. So how do I sort of follow up on this huge history of people studying this particular sea star? And the main reason why I really jumped into this is that when I came up here to the San Juan Islands, and for those of you who are here or who have been here and have seen what it's like out in the intertidal, you might remember or might notice that we don't have these really big mussel beds, but we still have Piezaster. We have quite a few Piezaster actually in the intertidal around the islands, but we have no mussel beds up here. So that got me thinking, what is going on? What is the sea star doing in a situation where sort of the number 2 main character is not here? So are they still having some sort of keystone predation or keystone species role, given that mussels are not in this area? And this is really what kicked off a lot of my dissertation work. Now, in order to figure out how they're affecting species in the intertidal, I needed to know who they were affecting first. So this gets to my first story that I want to tell you guys today, and this is getting into the diet of Piezaster in the absence of mussel beds. So I established some sites here on San Juan Island, and this was 2 years after sea star wasting had come through the islands and removed many of our Piezaster, but I was able to find 3 sites that still had a relatively decent population. And I started going around and looking at what they were eating. And one of the really nice and easy things about sea stars is that they eat externally. So this picture here is a really nice illustration of its stomach, that it's pushed out of its mouth, digests its food outside of the body, and then it takes that stomach that's now full of all those new nutrients and pulls it back inside. Which means when you're going out to see what they're eating, it's very, very obvious. I don't need to look inside my star to see what it's eating, because it's eating everything externally. So here's some nice examples here of a star that I flipped over that was eating a chitin. And when I pulled the chitin off, you can see where the flesh, where the mantle is being dissolved away. And then this I just had to add because it's wild. There's no way that that sea star caught a live crab. I feel like that crab either was dead or just walked into the mouth of a sea star. I'm not sure, but that was just too crazy to not share. So I started going out to my sites, and all 3 sites shared a couple really key characteristics. The first is that at a pretty low tide, about -1 meters mean low water, there's a really thick algal and kelp zone, and it is so thick that it's just too hard to sort of peer through all of that and try and find Piezasters. So I set that as a lower observation limit for all of the sites. The other really interesting thing is this deep crevice that shows up at about 0 meters, mean low water. And this is what that looks like in practice. This is one of my sites, Point Caution. It's just up the fire trail from the labs. And right at about 0, there's this nice deep cut that goes all along the rocks. And there's actually another one that goes up here. And then if you zoom into some of those crevices, this is where you start seeing Piezaster hanging out on these rocks. So we started going out at low tide and flipping over stars and taking a look and seeing what they're eating. And we did this a couple days, several days, more days, and we were finding maybe 2 or 3 stars feeding. And as I was going, I was thinking, you know, there's no way that I can make a diet study in the span of a PhD if I can't find stars that are eating. There's got to be something else going on. So I brought up this concern with a good friend and colleague who I very highly respect, who had been doing research in the intertidal for quite some time at the Friday Harbor Labs, Hillary Hayford. And she very gently reminded me that stars are not just existing out of water when it's convenient to me to go out and look at them in the intertidal, but they're also living when the water is back over them and it's nice and cool and maybe more tolerable at the time. So we started going out snorkeling instead. So when the water was a little bit higher, and what do you know? Up went all the stars up on the rocks, moving around, munching around, and suddenly I could find data about what they were feeding on. But just that pattern of moving alone was something really interesting that I knew that I needed to start taking note of. So here's again that same picture. Here's my RU from last year, Chris peering into the crevice to take a look at some of those stars. And here he is again at the same exact place, snorkeling at a higher tide, looking at those same stars to see what they're feeding on. And here's just sort of a splash. There's a couple quick things just to note that sort of jump out. The first being that nice tall bar right at the sort of conditions of the star is underwater and it's out of the crevice. It's sort of moved up on the rock face, and they're eating just so much more during this period of time, and sort of eating at a lower level, a lower frequency of eating in any of the other conditions, in water, out of water, in crevice, out of crevice. So that's the first thing that jumps out. The second thing, which takes a little bit more to piece out, is, well, first that barnacles, when they're feeding out or in water, barnacles is the main part of that diet. And what I like to think of when I'm thinking about this is, if you're in your kitchen and you're hungry and you want to eat something and you have a bag of chips on your table and you've got meals in the fridge, while you're sitting there thinking about what you're going to eat, you're probably going to grab that bag of chips and start snacking a little bit while you're waiting. And so these stars are sort of moving around looking for food, and while they're looking for food, these little sort of not very nutritious, not very fulfilling little snacks are scattered about. So they're just sort of slurping them up as they go. And then once you finally get to that meal, you finally find that thing that you want, or you reach into the fridge and you grab the thing you're going to make, it's going to take you a while to get through that. And so sea stars, as I was saying before, they're sort of slowly dissolving their food. So if they find that meal and they get to sit down and start eating it, there's a pretty good chance that the tide's going to go out before they're done eating it. So this sort of low tide, but they're still eating, tends to be these larger organisms that probably they caught while the tide was still high. It just took them longer to eat it. But we didn't want to just sort of, suppose that was happening. It's a nice story. I think that's what's happening, but we wanted to try and get some really nice quantitative data to show, to really show that that's what's going on. Which brings me to the second story on movement. And a lot of this project was headed off by Chris Gendry, my RU from last year, who's just a phenomenal human being. We decided to put cameras out in the intertidal. We wanted to have the cameras far enough from the rocks that we could watch the sea stars come out of the crevice as the tide came up, move around, track their movement on the rocks as they ate, and then watch them head back into the crevice. And hopefully maybe catch one of these elusive, finding a chitin and taking a whole day to eat it situations. So we got our GoPros, we got a whole bunch of bricks to make sure we weighed it down, because in the intertidal you get boat wakes, you get waves, and it's really challenging to keep a camera steady in the intertidal for extended periods of time. And this is Chris trying out the intervalometers on the GoPro. So we put everything in the field, we had everything figured out. And we took our first set of data and we got back this, which you can never plan for everything is basically the moral of that one. This is just stuff, particles. I mean, we live in a place that's highly productive. The intertidal is very turbulent. And there's just a bunch of stuff in the water. And since we're very shallow, the sunlight just lit everything up. So we slowly started moving the camera closer and closer to the rocks to figure out, okay, how close do we need to be in order to really get a good picture of these sea stars moving? And it turns out that you have to be really, really close. Which means ultimately we sacrificed our ability to watch the sea stars head far up on the rocks and actually watch them feed. So instead what we were able to get is this sort of movement in and out of the crevice. So as the tide comes up, how long does it take a star to start moving? How quickly do they move? Where are they moving? Are they making a beeline for more foody areas? And to sort of take into account this 3D nature of the crevice, we assigned these areas on the rocks different zones. So 2 and 3 being inside the crevice, 1, 4, and 5 being outside the crevice. And here's what that ultimately looks like, with 1 being out front, 5 being the top front, and 2 and 3 sort of being inside. So we took several days worth of data, looking at these stars, looking at their movements, watching them head in and out of crevices. And Chris took all of this data into ImageJ and manually tracked the movement with their central disk, sort of in every frame, clicking where the central disk was, and then using that to calculate distance and speed of the stars. And here's just a lovely example of one of those videos. This is a video. It's 1 minute or one photo that was taken every 2 minutes. And we've sped it up so that it's only 30 seconds long, but it actually spans 5 hours. And then the sun went down. So there's moving, there's sort of dancing. We were, you know, unable to tell whether things were actively eating at any point or if they were just hanging out. But at the very least, we were able to get a lot of sort of movement and velocity data out of that. And here's ultimately what that data looked like. And there's sort of a lot going on, but I'll walk you through some of them. First, again, this really tall bar graph. This was 2 video frames of a single star right as the sun was going down, and it just booked it up the side of the rock. So that was sort of a one strange star. But overall, the overall pattern that really fell out here is that stars are moving faster. When stars are moving, they're moving faster during a flood than an ebb. So perhaps you've been out of water for a pretty long time and now you're starting to get hungry. That water comes in and now it's time to go and eat. And perhaps you've been moving around for a while, so you're moving back into the crevice, or moving during an ebb tide a little bit slower. The second thing is that they're moving faster outside of the crevice than inside. So, and this is not including zero velocity measurements. So there's a lot of time where they're just sitting and hanging out. But when they're moving inside the crevice, it's slower. They're sort of hanging out in one area. And then once they hit these edges, sort of the 4 and 1 zone, they just head out. So this was just the start of trying to describe how sea stars are moving around. What are they doing, particularly as the tide's coming up. Our follow-up experiment that we had started but hadn't quite gotten to finish was also tethering foods. So we set up a little station here, a little pile of Z-spar with an anchor point where we tied off a variety of different foods like mussels, nucella, and a couple other things. And ultimately, we wanted to see if the speed at which the stars moved towards the food might be different, and whether or not we could use that as a proxy for food preference in the field, if they were sensing something that they wanted to get to. We ran out of time on this one, but it's open for folks to grab onto, or for maybe us to revisit at some point. And before I move on from this project, there were a couple other things that sort of came up as I was thinking about this issue with sea stars moving as the tide's coming up and down. And the main thing that sort of jumped out at me is, as the tide's coming up and the water is sort of hitting the sea star, you know, you're going from a warm situation to a nice cold situation. It could be temperature, it could be salinity, it could be something along those lines that's sort of telling the sea star that now it's time to move and it's time to go get food. But what's triggering sea stars when the tide is ebbing? So as the tide's coming back down, the sea stars are heading back into the crevice before they're fully exposed out of water. And this is something that I'm just throwing out into the universe for someone to take a look at and for someone to start thinking about. It could be something like pressure, but I don't think that that's something that's been looked at. And you know, these stars are doing this every day, all time of the year, when the tides are at different times of the day, so it's not light. So what's really driving this movement up and down, and how are they avoiding being stuck out of water so often? Lots of things to think about with sea stars. So this brings me to my third story, and this is going to be completely different than anything I've talked about thus far. And this is something that I've gotten into much more recently, ecomorphology. So this is looking at the organism's role ecologically. So what are they doing in the field, and does that match some of their morphological traits? So can we take a look at a star and say this is probably what it's doing in the field, or what it's eating, or whether or not it's a voracious predator or something like that. So for this particular project, we wanted to focus more on the skeleton and the ossicles. So all echinoderms, sea stars, sea urchins, sea cucumbers, sand dollars, all of them have an endoskeleton that's made up of calcium carbonate plates and spines. And these plates and spines are called ossicles. So this picture here that I'm showing is a variety of ossicles that you would find in sea cucumbers. And the shapes are just wild. You've got these sort of waffly shaped ones. This one reminds me of a cereal that I can't remember what the name of it was. You've got these hooks, you've got this wheel structure. The structures are outrageous. And in sea cucumbers, often you can sort of digest away some of the body wall and look at the combination of ossicles, and from the combination have an idea of what species you're looking at. With sea stars, they've sort of taken a slightly different route. They've piled in a whole bunch more ossicles and organized them in a way that creates a nice rigid structure into an actual skeletal structure, whereas sea cucumbers have them just sort of scattered about the body wall. Urchins take that a step further, and they fuse all of the ossicles together into something that's more like a test, where it's one solid piece. But the sea stars take these and they come up with all different shapes. They have flat ones that they fuse together into plates. There's round nobules. There's sort of these smaller mesh-like ones. And all these different species have all this different organization of these ossicles. And so I was really interested in what these skeletons looked like, first of all, across all of our different species that we have here locally. And not only what they look like, but how is their organization different, and does that relate back to what they do in the field, what they eat, where you find them, things like that. So we used some tools that were already here at the labs, thanks to Dr. Adam Summers. This is a micro CT scanner that's here at the labs and available to use for folks that come up here and bring their specimens up here. So I was able to take some of the stars that I had been collecting, and these are dead, these are fixed. They had been sitting in ethanol for a while, and we added tags to them, wrapped them in cheesecloth, although I think they have a more updated version now. It just shows that I haven't been CT scanning in a little bit. And we put them into these 3D printed cans that fit into the CT scanner, and Adam refers to these as the burritos. So I make a nice star burrito, and we place that into the CT scanner, and the CT scanner is taking hundreds to thousands of images of the one can, and it's just taking pictures at all different angles to capture sort of all the different parts of what you just put in there. And then you take these individual photos and you're using software to basically paste them all together into a 3D reconstruction of the thing that you just took an X-ray of. So here's sort of what that looks like. Here you've got one of the more raw images of the stars in the burrito, and then here's what it starts to look like when you reconstruct it. And you can see that it's all squished together. I scanned a whole bunch of stars all at once. You go through a series of more softwares, and you can eventually start to segment out these different individuals and end up with something stunning like this. So this is Dermisterius imbricata. This is the leather star, and all that you're seeing here is just the ossicles and the organization of the ossicles of the skeleton. So you've got these nice fused plates along the sides. You've got this more sort of spinuous type of ossicles on the inside. And then you've got these thicker, deeper ossicles that align the ambulacral groove. This is where the tube feet live. And then you've got these nice big spikes right in where the mouth and the stomach are. And we can zoom in and take a closer look at how this is all organized. But just right off the bat, we were sort of thrown back by how pretty this was. And so my collaborator and I sort of put this all up on a poster. We had scanned a bunch of different species at this point, and we took it to SICB, the Society of Integrative and Comparative Biology conference 2 or 3 years ago now. And we had a third person approach us looking at these photos, and he was like, you know what would work really well in terms of analysis for these guys? And he took us on a turn into fractals, which I will admit is completely new to me. I know some people shaking their heads. Math can be a little terrifying sometimes, but fractals, we're going to walk through them. I've had a lot of fun trying to learn fractals and wrap my brain around them. So hopefully some of this will help and we'll all get on the same page. So a fractal, this was one of the more simple definitions that I could find, is an irregular geometric object with an infinite nesting of structure at all scales. So one of the first things that sort of comes to mind, or one of the first things when you Google fractals, is this lovely triangle. So a nesting structure that is just nested within itself. It's the same pattern again and again and again. This is an infinite loop. Another example of that would be something like this, where you have something that branches and you have that same structure happen again and again and again and again and again. And then you end up with something beautiful like this dichotomous tree. And while this seems very theoretical and very sort of abstract, it turns out that you can find fractals everywhere in nature. And as far as this pattern right here goes, this was the first thing that popped to mind. This is a basket star, and this is something that we find here in our waters quite often. And actually we have 2 down in lab right now. And if you take a look at the pattern of their stars, you've got this dichotomous split, which then becomes another split, which then becomes another split. And it turns out that basket star arms are fractal patterns, which to me is just awesome and wild. So it doesn't surprise me so much now when I look back at our scans that someone who knew more about fractals sort of saw this and said, you know, there's potential there for this. So using fractals in terms of applying fractals, you get into something called the fractal dimension. This is the number that you're calculating when you're talking about fractals. Fractal is the repeating pattern. Fractal dimension is this number that you're calculating. And the fractal dimension is the ratio between the ratio of the detail of your pattern and how that compares to the scale at which you're measuring it. So here's a really nice example. This is where the first people thinking about fractals realized that this was something that could be measured. Benoit was measuring the coastline of Great Britain, and he realized that if you used a 200-kilometer scale to try and measure out Great Britain, that you would use a certain number of these and get out a certain length at the end, so 2,300 kilometers, which is sort of a rough estimate. You're missing some bits and pieces, but you're getting the general idea. So then, if you use 100-kilometer segments instead, you're now using more of them, and you're getting more of these sort of nooks and crannies and twists and turns, and overall your length ends up being larger. So now we're at 2,800 kilometers. If we do it again, we're capturing more nooks and crannies, we're getting a better idea of that overall shape, and our length is getting even longer. We're now at 3,500 kilometers. So he realized that the length of your thing is relative to the scale at which you're measuring it. And this is where that's sort of the baseline of the fractal dimension. Your length is always going to be relative to the scale at which you're measuring it. And how the relationship between how the length changes as you change the scale is your fractal dimension. Lewis Fry Richardson took this a step further into the like brain exploding zone of if your length is getting larger every time you make your scale smaller, that means that every length is ultimately infinite. So as you keep measuring at a smaller and smaller scale, your length will always be getting larger and larger and larger. So all of the lengths that we measure is relative, and ultimately any length that you measure is infinite. So the next time someone asks you, how tall are you? You can say, I am approximately 5'9", but mathematically my height is infinite. It was one of my favorite things as I was learning about all of this. I just had a whole afternoon of like, can you believe it? We're infinite. Okay, so back to how you actually apply this besides infinity. Here's an example that I worked out for what this actually looks like when it comes to the analysis. And this is called the box counting fractal analysis. Let's take our urchin as an example here. Step one of the fractal analysis is to make a bounding box. So you make a box that is no larger and no smaller than your organism. The next step is to say, is your critter in the box? We have one box. The organism is in that box, so the answer is yes. If we started plotting this with the x-axis as boxes available and the y-axis as boxes occupied, then this would end up being 1 and 1, all set. Step 3 is just to divide. So this is where the fractal pattern starts coming in, right? A pattern within the pattern, within the pattern, within the pattern. So we divide it. Now we've got 4 boxes. Is it in your box? 1, 2, 3, 4. Yes, 4 for 4. We're set. We divide it again. Is it in the box? Yes, it is. We divide it again. Is it in the box? And now we have an issue. Now we're starting to reach some areas where your organism is no longer taking up that space. And so you have, so we went from 1, 4, 16, 64, 256. As your boxes available continue to increase with that squared function, the boxes occupied is going to start to slow down. It's going to start to peter off. And you just keep doing this iteration after iteration after iteration, and eventually your line is going to start to reach sort of an asymptote. So boxes available will continue to climb, boxes occupied slows down in the number that they're filling. And that's because you're getting into that detail and you're really capturing the outline of your thing, whatever you're measuring. In this case, it's an urchin. So if we take that graph, boxes available by boxes occupied, and we log both axes, you end up with a straight line. The slope of that straight line is dependent on that relationship that was happening up here. So how much of your creature was taking up the boxes available. The slope of that line is the fractal dimension, and that's it. That's how the box counting fractals work. So if you take something like that big, sharp, spiny urchin and you compare it to something like our purple urchins with little tiny spines, you end up getting a larger fractal dimension in this urchin than you do in this one, because this urchin is taking up more space within its box than this urchin was taking up in its box. So a fairly simplistic explanation of how it works, but we can take this and apply it to something more complex, like this star. This is the slime star, and it's got, spines on spines, and it's got like Swiss cheese holes all through it, and this nice big gap all along that mouth region. And we can apply this same calculation to take a look at how complex this skeleton is compared to like the dermasterius that I showed you earlier. And much of the more math heavy portions of this collaboration is all in thanks to my collaborator and very good friend, Cassandra Donatelli, who is up here as an instructor for the fish class right now. Ultimately, she took what was traditionally a 2D analysis and transformed it into a 3D analysis so that we could take that sort of 2D explanation that I gave you and apply it to something like this, a 3D sea star, and take into account not only that it has spines, but the spines are going in different directions and the inside of the star is hollow, and there's just a lot more going on. So that's one of the big directions that we're taking these scans, but there's a lot of other stuff that we can do with them. This is a beautiful example of just some of the species that we have here. So the slime star, we've got a solaster, this is Pycnopodia down here, this is 3 Henricia blood star individuals, Piezasters here, Episterius here, Leptosterius here. And this particular picture has them color-coded by density of the ossicles. And this isn't density in terms of the number of ossicles, it's density in terms of what the material is that the ossicle is made out of. So you see things like the tips of arms being extremely dense ossicles, areas along the ambulacral grooves having more dense ossicles than other places in the body. So we can look at density, we can look at the ossicles themselves, how many are there, what is the size. We can start measuring all these different traits, and ultimately what I'd love to do is map a lot of these different morphological traits on a phylogeny, and start bringing in some of these things that we know about their ecology, you know, which ones are sponge eaters, which ones are filter feeding, and can we see that in their skeletons? Can we see these differences in the way that these are organized? And that's where this work is headed. We're still scanning a lot of stars right now. I've got a whole set of stars lined up to head into the CT scanner once the fish class is done, and hopefully you'll be seeing more of this stuff coming out soon. So with that, I'd like to thank, there's always too many people to thank, my advisor, Jennifer Rusink, who's given me lots of flexibility to play around with all the things that I find interesting, and also figuring out where to sort of rein me in and make sure that I stay focused. Chris Gendry, my RU from last year, he just graduated and he's looking for a job. So if you need a hardworking and absolutely fantastic, phenomenal person in your lab, I'm happy to share his contact info with you. And then just a lot of other folks. Thank you so much.
That was wonderful. Thank you so much, Mo. That was a very approachable explanation of fractals, which is a hard thing to do, and you did it so well. So we'll take questions now, and we can do what we've done in other weeks because we don't have a hand raising function. If you have a question, if you could just go ahead and type question in the group chat, and then I can unmute you and you can ask your question of Mo.
Oh my gosh, honeycombs. Yes, thank you. I was just looking at the chat. Mm-hmm.
Okay, it looks like John Allen has a question. So John, I'll go ahead and unmute you.
Hey Mo, that was awesome. Thank you.
Thanks, John.
So what I've noticed in super artificial lab environments is that Piezaster eat a lot more when I turn the lights off at night. So have you done any, or did Chris do any observations? Did you do any night dives, or do any, set up any infrared cameras, or do you have any intention of doing that? I'd really love to know what they're doing at night, whether the tide is in or the tide is out.
Mm-hmm.
I think that'd be really fun.
Yeah, we have not. I would love to know what they're doing at night. The other limitation that I didn't mention is that we're also not looking at them at really high tides. We're sort of limited to medium tides because we did all of this work either walking out at the intertidal or snorkeling. And at some point the tide gets so high that you can't snorkel down far enough. I can't snorkel down far enough to go see the stars. So we didn't have really high tide, and we don't have night stuff. And I think it would be so cool to see both of those. And I think you hit the nail on the head. It would require diving and some sort of infrared cameras, which I would need to get my hands on to take out there. But I suspect they might be eating quite a bit more during that time as well. Although I don't know how much they're moving around when they're out of water, even at night. Great question.
Great. It looks like Amy has a question. Uh-oh, did we lose Amy?
Move my chat box over here so I can see what's going on.
Maybe she'll come back. Looks like Billy has a question. Oh, did we lose Billy too?
I'm here.
Okay.
Can you hear me?
Yep.
Oh, great. That was cool, Mo. It's really interesting. I love the fractal part and the beautiful skeletons. So getting back to your original question about whether you think that they're acting as sort of keystone species, do you think that they are, like say, keeping down the barnacles enough or whatever so that there is more diversity in the area where they are, or do you think it's just a very different system because they don't have the mussel beds?
Yeah, so I started another project to try and get at this a bit more, which included exclusion cages in the field, We put these out last year. And the idea was that if we could keep Piezaster out, that the things that maybe they've been eating and impacting would have a chance to grow within the exclusion cages. I don't have any data on this, but just having sort of visually looked at these plots over time and just what I know that sea star are eating out here, I kind of suspect that they don't play the same role. I don't think that they are a keystone predator in the San Juans the way that they are in the outer coast. The barnacles are not out competing other things in the same way. There's not that sort of pressure to open up space as much as there is when those mussels come in. So I think that Piezaster are just sort of a general predator here, just sort of mowing as they move along, but not having this massive impact. Yeah.
Cool. Thank you.
Mm-hmm.
All right.
Looks like Flo.
Yep. Let's go ahead and unmute Flo.
I just love the breadth of your work, Bo. And it's absolutely fabulous. And one of the things I was thinking of, would it be possible? What do you think about creating a situation in the labs where you could control the tidal flux, and you had stars, and you had prey? What do you think about that?
Yeah, I had this sort of dream conversation with someone the other day about building this sort of mock intertidal with sort of a tall rock with a nice deep crevice near the bottom, and I could just recreate this whole system with the tides coming in and out. It would be amazing. So then I could actually watch individuals, which sort of brings up another thing, which is that tagging stars is extremely challenging and really hard to do. So I haven't been able to follow any individual stars in the field. Which is why I was hoping cameras would work. But if we were able to set up something in the field or in the labs like that, you would know which individuals you were watching and how much they were eating and how much they were moving. And yeah.
But you might even be able to answer some of your simple questions about anticipation and rate of movement and some of the other things that are just the behaviors that underlie the complexity of their response in the intertidal, which is a much more complex habitat.
For sure. I absolutely agree. I think it would be, we could potentially get that type of information in the lab. The only reason why I hesitate at all is just that sea stars are not known to do what you want them to do when you ask them to. So I tried to do a diet study in the lab just to feed them different things and see what they would eat, and I couldn't get them to eat for about 2 months. So they can be very picky about what they're doing, and especially when you bring them into a lab setting, they seem to act a little bit differently than they would in the field.
But I'm also thinking that providing that tidal flux might be important to motivating their behaviors. And so if they're just in a tank in the lab, that might not be an appropriate replicate.
That's a very good point. Yes.
Anyway, it's a wonderful talk, both. I've been wondering about all of this stuff that you're doing. It's just fabulous. Sea stars are just incredible organisms. Yeah. Moving slowly.
Very slowly, but fabulous. Yeah.
All right. It looks like Amy has a question, so let's pop back to her. And Amy, I'll unmute you so you can ask your question.
Great, Flo. That was a great talk.
Thank you.
So I was wondering, are the sea stars here where they're eating a bunch of barnacles and just a few larger things, are they any smaller than the sea stars where, on the outer coast or wherever, where they're, they've got a bunch of mussels that they can feed on?
Yeah, so I only know sort of anecdotal answer to that, and I would say yes. I have lots of measurements from my stars up here, and I've got photos of the stars that Bob was looking at, especially like that first photo. Just looking off that first photo, I would say yes. His stars are 2 times larger than mine, at least, in that photo. But I also haven't been out there recently, so I don't know if the size that I'm seeing is because of sea star wasting, so it's smaller stars that are coming back from that, and maybe we're seeing that on the outer coast too, or maybe there's an actual difference. I suspect that there is. I don't think that they're getting quite as much nutrition here on barnacles as they are on mussels in the outer coast. I think they're much bigger out there, but I need to go do some actual measurements.
Oh, great, thank you. All right, I think our next question was from Trish. So Trish, let me unmute you and you can ask your question.
Thank you. I still remember when you gave that paper, and I still have the sea star that you gave me at that meeting. I was so excited, Mo, about that. You remember that, I'm sure.
Oh yeah. Oh, yeah.
And what I'm so pleased to see is that you've continued this. I think there's a story in there on the heaviness of the and the distribution of these forms as to the strength of these. I don't know how it can be measured, but for the way those fish people measure everything this way, that way, and the other way,
Yeah.
I think you could do that with the stars. And
yeah.
I hope you save me a picture of that whole group you have. I'd love to get a copy of that.
For sure. I mean, it's been, I would not have been able to break into this field without Cassandra and some of the other folks in that fish lab. They have definitely been holding my hand through most of this and helping me learn just morphology and the world of morphology in general. So, yeah.
Terrific.
All right, it looks like we have a question from David, but it looks like maybe he disappeared. So let's go to Pierre, who is now unmuted, maybe.
Hi, sorry.
You're good.
I'm up here in Victoria, and I just want to say it's been awesome to have access to these seminars. Like, just really, 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 really cool.
We're glad you could make it.
Yeah. I wanted to ask you about the QR code that was on the image with all of the different species.
Yeah, so that was, we generated that for one of our posters at one of the sick bees, and that's just a link to more, went a little too far, to more 3D models, to GIFs of sea stars spinning, of the models spinning, if you wanted to see more of them.
That's really exciting. It's interesting how just the variation of density.
Yes. Some of this, so this one was by far the most interesting, and this is one of the reasons why I want to revisit the density question. This is three individuals of a species complex, so they might be the same species, they might not, but they live in similar places and do similar things, and yet we're seeing kind of a big difference in density of the ossicles. So I want to revisit and first of all just make sure that we sort of did that properly, and try and figure out why this type of pattern is coming up. What else is happening that's driving this really big change in ossicle densities?
It's interesting because I'm sure just by the nature of fractals, like the further in you zoom in, the more you end up seeing. In my marine invertebrates class up here, we got the chance to actually use the scanning electron microscope on the ossicles of sea urchins, and even no matter how close we got, it just seemed like it was endless.
Right, there's more. There's like porous, but it's still, it still has a pattern to it. It's wild. Yeah. So
it seemed like the limit was almost like the gold plating particle that we had put on. Like I don't even know how you would go about measuring that, but hearing you talk about fractals is like super exciting because now I have all these ideas in my head.
Yes.
Thank you.
Yeah. We're gonna also go through, so these star scans take quite some time to do. So we're gonna take smaller snippets of these and do a much higher resolution scans and see sort of how far we can get and see how much detail we can see. Yeah.
I'll be super excited to read about that. Thank you.
Yeah, thank you.
All right, I'm looking through the chat. You've got some offers of new collaborations, so definitely have a look through that when you get a chance.
I'm here for it.
Your lab is watching from Willapa, so that's lovely.
Thanks.
They think you're amazing. I agree. Okay, and it looks like the lab has a question which says, do stars that move fast, like Pycnopodia, have fast-looking ossicles?
This is such a good question. We haven't looked that closely yet at what the individual ossicles look like, but what I can tell you about Pycnopodia, and you can sort of get this from this picture here, but they have totally reduced the number of ossicles that they have to the point where they're not using their ossicles for a rigid skeleton. They're almost entirely using hydrostatic skeleton for their shape. If I had to guess, I would say that one, it makes them more flexible in terms of getting things like urchins, getting between the spines. If you don't have as rigid of a skeleton, you can sort of wiggle through smaller spaces. Second, it might make them lighter in terms of being faster. They're not weighed down by their endoskeleton. And well, those are my big two thoughts on that. But they are one of the fastest sea stars, and I think a big part of that's just the pure number of tube feet that star has. But they're way more flexible than any other star, and that probably plays a role in that. It's a great question.
It looks like Flo might have another question, wondering about the density or size has something to do with the ability to fit into the size structure of the habitat.
Yeah, so that's sort of getting back to the Pycnopodia. They can, just from that lack of rigidity in their skeleton, I suspect they would be able to get through much smaller, tighter spaces. That being said, all of the stars, even with this rigid skeleton, are all fairly flexible. If you pick up a rock and you find a star that's sort of been half flattened underneath, they totally just bounce back from that. And I don't know how they're doing that. So I suspect, and I'm hoping that as we get tighter in on these sea star scans, what I suspect is that the ossicles might not actually be fully connected, and that gives them a lot more ability in order to bend and move. So if they are sort of wiggling under a rock, that they can just move all of those ossicles, sort of flex them in whatever direction they need to get through whatever they're trying to get to. But they can get through some small, tight spaces regardless, it seems. Yeah, wild. I know, sea stars are crazy.
I think we're out of questions. Lots of cool things happening in the chat, so definitely read through that. I just want to ask one more really quick question. I know you have to have stars of a certain size to be able to put them in the CT scanner. Do you think anything like the density, for example, might change across ontogeny, or is there any, how can you test that if you're limited by your size of the burrito?
That's a great question. So right now, what Katie is referring to, the CT scanner is only so large, and we can only scan a certain size star if you're talking about the full organism. And right now, it's limited pretty much to the size of the palm of your hand, which means like this Pycnopodia was a pretty small Pycnopodia, and that goes for all the rest of them. There's two options that I can think of here to look at that. One, which sort of Cassandra already said in the chat here, is that you just look at arms, or you just look at a portion of the star rather than the whole individual. The other option is that we have a sea star species here that only gets so big, and if I could get or grow some or something in the lab, and we end up with having this sort of size variation within this particular species, and I'm referring to Leptosterius, the 6th grade star that really gets to the size of like a $50 at most. it's a pretty small star. So if we can get a whole series of Leptosterias through their age, we could look at whole individuals rather than just the arms.
Awesome. Sounds like you've got plenty of other things to keep
I've got
working
like
on.
5 more PhD planned.
You'll be busy. Yeah, no, I think you'll stay out of trouble because you have so much to work on.
Yeah.
Well, thank you again, Mo. This was a wonderful talk. Such compelling work. You explained things so clearly. Thank you so much.